In this tutorial, we walk through the sample preparation steps required for the nCounter microRNA expression assay. All of your experiments should be designed in sets of 12 assays, and all reagents are supplied in 12 reaction single-use tubes. Therefore, the protocol described in this tutorial is for one set of 12 hybridization assays. Before starting the sample preparation protocol, your RNA samples should be normalized to the same concentration, about 33 nanograms per microliter, using RNase-free water. The microRNA sample preparation kit is shipped in 2 boxes. One box, which should be stored at -20 degrees C, contains the nCounter miRTag reagent, the microRNA annealing buffer, PEG, the ligation buffer, ligase, and the ligation cleanup enzyme. The second box, which should be stored at -80 degrees, contains the microRNA assay controls and the capture probes and reporter probes of the microRNA code set. Thaw a tube of each of the reagents stored at -20 degrees on ice. Also, thaw a tube of the microRNA controls on ice, but do not thaw the tubes of the capture probes or reporter probes at this time. Make sure your PCR machine has been programmed with the annealing protocol, the ligation protocol, and the purification protocol. For details of each of these protocols, please refer to the printed microRNA sample preparation manual. To begin the protocol, prepare a 1 to 500 dilution of the microRNA assay controls. To do this, we recommend adding 499 microliters of water to 1 microliter of the microRNA assay controls in a sterile microcentrifuge tube. Mix well and briefly spin down to collect to the bottom of the tube. Place the tube in the ice bucket. Prepare an annealing master mix by combining 13 microliters of annealing buffer, 26 microliters of nCounter microRNA tag reagent, and 6.5 microliters of the 1 to 500 assay controls that you just made. For all steps of the sample preparation protocol, it is recommended that you mix each reagent gently by flicking on the sides of the tube and spin it down in a picofuge just prior to adding it to a reaction mixture. Do not vortex to mix the samples at any point, and do not use a high-speed microcentrifuge to spin down the samples, as your code set might be spun down to the side of the tube. Mix well by pipetting up and down. Spin briefly to collect the material to the bottom of the tube and store in your ice bucket. To one of the strip tubes supplied with the kit, add 3.5 microliters of the annealing master mix into each one of the tubes. Next, add 3 microliters of your normalized RNA sample to each tube. Cap the tubes and flick the tubes gently to mix and spin down briefly in a picofuge. Place the strip of tubes in the thermal cycler and begin the annealing protocol. While the annealing protocol is proceeding, prepare a ligation master mix by combining 19.5 microliters of PEG and 13 microliters of ligase buffer. Please note that the PEG solution is viscous and should be pipetted slowly and carefully to ensure accurate transfer of volume into the master mix. Mix the tube well by pipetting up and down and spin briefly to collect the material to the bottom of the tube. When the annealing protocol is finished and the temperature is holding at 48 degrees, add 2.5 microliters of the ligation master mix to each tube. Do not turn off the thermal cycler. You will need the block to be at 48 degrees in later steps. Flick the tubes gently to mix and spin down in the picofuge. Then return the tubes to the thermal cycler and incubate at 48 degrees for another 5 minutes. For the next step, it is extremely important that the tubes do not get removed from the thermal cycler. Maintaining the temperature of the tubes at 48 degrees is critical for optimal assay performance. Carefully remove the caps from the tubes, leaving the stripped tubes in place in the heat block. Add 1 microliter of ligase directly to each tube while the tubes continue to incubate at 48 degrees. There is no need to mix at this point. Again, the tubes should never be removed from the heat block during this step. Immediately after addition of ligase to the tubes, recap the tubes, leaving them in the heat block, close the thermal cycler, and initiate the ligation protocol. When the ligation protocol is complete, the tubes can be removed from the thermal cycler. Add 1 microliter of Ligation Cleanup Enzyme to each reaction. Flick the tubes gently to mix, and then spin down in a picofuge. Return the tubes to the thermal cycler and begin the purification protocol. When the purification protocol is complete, add 40 microliters of RNase-free water to each sample tube. Mix well and spin down in a picofuge. If necessary, the samples can be stored at -20 degrees for up to several weeks before hybridization.